Assalamu alaikum. Very good afternoon, ladies and gentlemen. I warmly welcome to all of you on the behalf of organizing committee on the 8th International Conference on Applied Zoology 2025. Myself, Muhammad Qasim Raza, and I will be the moderator of the session, which includes talks from the fields of nutrition, genetics, histology, and entomology. Let's we start our session with the talk of Professor Dr. Saima Masood from University of Veterinary and Animal Sciences on the topic of histomorphological evaluation of curcumin ameliorative effect on nicotine toxicity in testes and epididymis in rabbits. Ma'am, please come and give us valuable remarks.
So, Bismillahirrahmanirrahim. I'm presenting here my abstract as histomorphological evaluation of curcumin ameliorative effects on nicotine-induced toxicity in testes and epididymis in rabbits. And myself is Professor Dr. Saima Masood, working as a professor tenured in the Department of Anatomy and Histology at University of Veterinary and Animal Sciences, Lahore. So, we know that cigarette smoking is identified as a major risk factor for the progression of reproductive health in individuals, and one of the pharmacologically active constituents of the cigarette is nicotine. And in this trial, we have decided to see the adverse effects of the nicotine on the male reproductive health, particularly on the testicular and epididymal tissues. And we wanted to see the effect of curcumin which is a natural compound, and it has many antioxidant and anti-inflammatory effects on the individuals. And we want to see the at cellular level how potentially it is active in mitigating such toxic effects on the reproductive organs of the male. And this study was aimed to evaluate all these things on the male rabbits. And for this purpose, a total of 24 New Zealand white rabbits were randomly assigned into 4 groups, and each group was having 6 rabbits. And this trial was having a duration of 35 days, and it was divided into 2 phases. Nicotine was administered to T2 and T4 groups for at least 17 days, and followed by curcumin treatment in T3 and T4 for the next 17 days. At the end, testicular and ductal epididymal tissues were collected after sacrificing the rabbits, and they were proceed for standard histological methods like using hematoxylin and eosine staining methods, and analyzed for under the light microscope for the morphological purpose. Here are the treatments. T1 was having control group, and T2 was nicotine-induced group at the rate of 0.75 milligram per kg body weight, while T3 was administered curcumin at the rate of 100 milligram per kg body weight. T4 group was having nicotine as well as the curcumin as an ameliorative effect. Here is the graphical representation of our different parameters like comparative mean values of the testis weight, comparative mean values of the testicular epithelial height, comparative mean values of the testis interstitial space length, comparative mean values of the epididymal epithelial height, and then comparative mean values of the epididymis interstitial space, as well as the comparative mean values of the epididymis length was measured. So nicotine significantly reduced testicular weight, which was restored by curcumin. Here are the parameters, elaboration of the parameters that we can see that induction of nicotine have significantly reduced testicular weight, which was restored by curcumin. And in T4 group, we can see that the partial recovery was obtained by administration of curcumin. And then in histomorphological presentation, you can see that these are hematoxylin and eosine stained slides at 100x. And we can see that the seminiferous tubules are present over here in the testicular tissue. And curcumin improved the epithelial height and interstitial space in both testis and epididymis. And epididymal epithelial tissue interstitial space was significantly reduced in the curcumin group, and significant difference was observed in epididymal height and also observed among the groups. And these findings are going to elaborate that how this curcumin is having a ameliorative effect on the testicular as well as the epididymal morphology at the cellular level. So it should be incorporated in as a supplement in the diet of the individuals for improving fertility as well as for improving the health of the individuals. Thank you very much.
Thank you so much, ma'am, for your kind information and your valuable remarks. Our second member of session is Dr. Muhammad Rehan from University of Agriculture, Faisalabad, on the topic of climate smart nutrition strategies for livestock, integrating fodder innovation and One Health. Sir, please come and give us insightful comments about your topic.
Bismillahirrahmanirrahim. My name is Dr. Muhammad Rehan. I am working as a lecturer at the Institute of Animal and Dairy Sciences, University of Agriculture, Faisalabad. Before to present my presentation, I am saying thanks to 8th International Conference on Applied Zoology 2025, who is organized this conference, which will help the scientists and the people who are working in this domain. They will collaborate and will work for the betterment of the Pakistan. I will also thank to University of Punjab and Government College University Faisalabad, who is in collaboration with this Applied Zoology Society of Conference. Before going to the presentation, I will tell you my presentation topic. The topic of my presentation is climate smart nutrition strategies for livestock, integrating fodder innovation and One Health. Before I discuss my presentation, I will tell you why I am presenting this climate smart nutrition strategies, because the background of my presentation is this presentation will highlight how livestock nutrition can be improved in a way that not only enhance productivity but also support public health and environmental sustainability. I will also discuss how climate resilient fodder system and One Health concepts can be combined to create a future ready livestock model. The background of my presentation is, we know that livestock in climate vulnerable regions, especially in developing countries like ours, face multiple challenges. The challenges, if I describe in one by one, the first major challenge is feed scarcity due to reduce animal growth, and second, the major point is productivity declines due to stress and malnutrition, and third, poorly feeding conditions increase the risk of zoonotic diseases. which can transmit from animals to humans. Therefore, we need holistic approach to address all these issues together. Need for climate nutrition, smart nutrition. To overcome these challenges, we need a policy framework that combines three important elements. First element is climate adaptive fodder strategies. Second is One Health principle. Then third is farmer resilience training. If we discuss the first element, which is climate adaptive fodder strategies, so that in this point the feed remains available even under harsh weather. The second point, which is main concern, One Health principle. In this point link animal nutrition with human and environment health. And third, which is most important in my point of view, is farmer resilience training, because we cannot go to the final solution if we do not do our farmer train, because solution must be practical and adaptive at the field level. Third, now we will discuss the next slide, impact of climate change on fodder. As we know that climate change has disturbed natural fodder cycles, seasonal grasses now grow irregularly, biomass yield has reduced, and nutrient content is often imbalanced, especially during heat waves or drought. We know that these are the major problems which our fodder are facing. This directly affect milk and meat quality and increase susceptibility to disease. Therefore, maintaining consistent nutrition has become a difficult. Next, proposed climate adaptive solutions. To overcome these issues, we can adopt innovative fodders such as hydroponic fodder, multi-cut draft resilience grasses, ensiled or fermented forages, and third and lastly, locally balanced rations. In hydroponic fodder, which grow without soil using minimal water, in multi-cut draft resistant gases. In this, we can harvest multiple times even in dry conditions. In fermented forages, we can ferment and preserve nutrients for longer period. Like if I give the example, sorry, for silage, haylage are the best examples for fermented fodder. And third, and locally balanced rations, which utilize available crop residues and supplements effectively. These strategies ensure year-round feed scarcity. If we discuss about all these issues in respect to the One Health perspectives, no, in One Health perspective, animal nutrition is directly connected to antimicrobial resistance and zoonotic disease spread. I will explain how this anti-animal nutrition is linked with antimicrobial resistance. As we know that poor nutrition, weakness, immunity, increasing dependence on antibiotics. This lead to drug resistance, which become a danger for both animals and humans. By improving nutrition, we can naturally reduce antibiotic usage, promoting safe food systems. And immune supportive feeding strategies, if we one practical solution is the use of nutraceuticals. In these strategies, one practical solution is the use of nutraceuticals such as herbal extracts, probiotics, omega-3 fatty acids, vitamins and minerals. These are naturally immunity boosters, which reducing disease incidence and lowering the need for antibiotics. In this way, nutrition becomes a disease prevention tool, not just an energy source. And the last immune supportive feeding strategy, one practical solution is the use of nutraceuticals. In this strategy, to adopt one practical solution is the use of nutraceuticals such as herbal extracts, probiotics, omega-3 fatty acids, vitamins and minerals. These are the strategies successful at immune booster, which reduce farmer disease incidence while lowering the need for antibiotics. In this way, nutrition becomes a disease prevention tool and not just an energy source. If we do not train our farmer, immune supportive feeding strategy, if we one practical solution is the climate smart nutrition is in this strategy about feeding animals, which is about the sustainability of life such as herbal protecting probiotics, health, and conserving the environment by combining these innovation. One Health perspective and farmer empowerment, which reduces disease incidence and lowering the need for antibiotics. In this way, nutrition becomes a farmer prevention. We cannot just final energy system. by combining photo innovation, supportive feeding strategies, one practical solution is the resilient and sustainable lifestyle system for the one practical solution is the use of nutraceutical. Thank you for your attention. Probiotics. I will be happy to take vitamin or injection. These are Allah.
Thank you so much, sir, for your penetrating analysis and useful advices. Our next member of session is Dr. Huma Ambreen from Government College University, Faistabad, on the topic of eco-friendly nanoparticle integrated hydrogels, a promising platform for advanced biomedical applications. Ma'am, please come and give us insightful comments and valuable remarks on your topic.
Bismillahirrahmanirrahim. This is Dr. Humam Brain from Department of Nutrition Sciences, GC University, Faistabad. The topic that I am going to discuss today is eco-friendly nanoparticles integrated hydrogel, a promising platform for advanced biomedical application. So we will start with it. When we talk about the biomaterials, these have gained importance in modern medicine because there is need for eco-friendly and sustainable solutions in biomedical fields, and where nanocomposites and hydrogels have taken special position. These hydrogels are 3D polymeric network with high water content, and they have higher biocompatibility and flexibility. And they have enormous applications and benefits in drug delivery, wound healing, and tissue engineering. When we talk about the nanohydrogels, these are the hydrogels that are designed in nano size, that is from 1 to 100 nanometers. And they are very minute, thus they can move easily within the body and target different types of tissues and cells. Similar to regular hydrogels, they are soft, compliant, and can contain great amount of water. When we talk about the eco-friendly approach, so mostly in our studies, we are using agro waste as raw materials, such as apple and carrot pomace. Similarly, we are using aloe vera gel, turmeric, and zinc nanocomposites that provide a sustainable and green synthesis methods. And these are sustainable solutions towards biomedical problems. Here are some of the hydrogels that we have made, and these have been done recently. One is apple pomace based hydrogel, and the other one is carrot pomace based hydrogels. These have been made in oral hydrogels that are edible, and these polymeric hydrogels combined with nanoparticles, and they have enhanced mechanical strength, stability, and biofunctionality. Here are some functional properties of these hydrogels, that they are more biocompatible, they are environmentally safe, they have controlled release capacity, and they have higher antimicrobial and antioxidant effects. These are some of the SAM images of apple pomace hydrogel that we have made. And these show that these hydrogels have porous structure. And however, these pores are interconnecting, and that help in swelling and water absorption properties. These are the images, SAM images of carrot pomace hydrogels. And these also show the same properties because they have plant fiber reinforced hydrogels. These are UV visible spectrometric analysis of these hydrogels. And here you can see the red line, which is representing apple pomace based hydrogels. And they have more of the polyphenolic compounds and flavonoids. And whereas in case of carrot pomace based hydrogels, which has been shown in green line, here you can see, and these have mostly carotenoids. These are X-ray diffraction analysis of apple pomace hydrogel, and the structure has shown that these are crystalline in nature. And similarly, here you can see uh, the apple pomace based hydrogels. They also showed the crystalline structure. When we talk about the biomedical applications of these hydrogels, nanohydrogels, they help in wound healing, controlled drug delivery, weight management. In our lab, we are doing all of these. And these also help in tissue engineering and regenerative medicine. When we talk about the wound healing application, as I have already told that these are important as anti-inflammatory agents, antioxidant agents, and antimicrobial agents. Therefore, in vitro and in vivo results have shown that faster they result in faster tissue regeneration, and there are reduced risk of infections. Similarly, such as I have shown you, apple pomace hydrogels, these help in weight loss, and because these help in appetite control, so they result in slow and controlled release of the bioactive compounds. They have basically controlled release mechanism. It means that they will control where to release the medicine or important nutrient compound. And this may be done with the help of controlled diffusion controlled delivery, pH enzyme responsiveness, and site-specific targeting. When I have already mentioned that, and these have good antimicrobial properties. And similarly, we have studied about the synergistic effect of turmeric, aloe vera, and zinc nanoparticles that helped in reduction in microbial growth in case of wound models. Here, as these are eco-friendly, so they have environmental benefits. They are helpful in agro waste utilization. So they help to reduce the pollution. Similarly, green synthesis methods have been used, so all these avoid the toxic chemicals. And these also these also provide the sustainable biomedical solutions. In vivo and in vitro studies have been performed, and in vitro they have been used in cytotoxic analysis. Biocompatibility has been confirmed, and the controlled drug release has also been confirmed. Whereas in case of animal models, they have been used in wound healing, and they help in positive tissue regeneration, and they also have been confirmed for the therapeutic efficacy. However, there are some challenges and limitations, and these include large-scale production issues, need long-term toxicity studies, and regulatory approvals are also a big issue. So we can conclude that eco-friendly nanoparticle hydrogel system provide multifunctional biomedical solutions. They have a promising factor for next generation biomedical applications. And however, there is need for interdisciplinary collaboration for clinical translation. There are some references, and with this, I will end my presentation. Thank you. Thank you so much.
Thank you very much, ma'am, to give us valuable information. Our next member is Dr. Sana Zahoor from Virtual University on the topic of optimization of fermentation parameters for maximizing fungal zinase production. Ma'am, please come and give us valuable remarks.
I am Dr. Sana Zahoor from Department of Biological Sciences, Virtual University of Pakistan. The topic of my presentation is Optimization of fermentation parameters for maximizing fungal silanase production. Silanase is an important industrial enzyme that breaks down xylan, which is the second most abundant polysaccharide in plant cell walls, into simpler sugars like oligosaccharides and xylose. It finds applications in many industries, like in paper and pulp industry. It is used for eco-friendly bleaching and for reducing chlorine use. When added in animal feed, it improves the digestibility and nutrient uptake or absorption. In textiles, it is used for the biopolishing and improving the textile or fabric quality. It is used in different food processing units, like in baking industry. It is used to improve the dough quality and for the clarification of juices. It is also used for the improved scarification of biomass for enhanced bioethanol production. There are different sources for silanase production. Among the microbial sources, fungi like Aspergillus, Rhizopus, Trichoderma, and Penicillium are potent producers of silanase. Different bacteria like Bacillus, Streptomyces, and Clostridium also produce silanase. Among yeast, Cryptococcus and Fiscia are reported to produce silanase. Among the different microbial sources, fungi are preferred for the production of silanase because they are capable of producing large amounts of silanase in the extracellular medium. They are capable of using cheap substrates for silanase production. They can use a wide range of substrates. The fungal silanases can work well under a wide range of pH and temperature conditions. In addition, the fungal silanases are better suited for harsh industrial conditions, and most of the fungi are generally recognized as safe. Two different methods are used for the production of silanase: solid state fermentation and submerged fermentation. Submerged fermentation is preferred because it is easier to control the process parameters like pH, temperature, and aeration in submerged fermentation. Submerged fermentation gives better microbial growth that leads to higher enzyme production. It is easier to scale up the submerged fermentation in industrial bioreactors. Since the fermentation conditions are consistent in submerged fermentation, it leads to consistent and reproducible enzyme production. It is easier to recover and purify the silanases that are produced after submerged fermentation. Now the methodology. Different samples like soil, compost, agro-industrial byproducts, wood residues, and decaying plant material were collected, and silanase-producing fungi were isolated and screened on xylan agar screening medium. The isolate giving the maximum zone of hydrolysis on xylan agar screening medium was selected and was characterized on the basis of morphology and molecular means. In morphological characterization, macroscopic characteristics and microscopic characteristics were observed, while the DNA was isolated and 18S rRNA gene was amplified using universal primers, and phylogenetic tree was constructed using MEGA12. For the optimization of silanase production, effect of different media, pH, incubation time, carbon sources, and temperature was studied on the production of silanase. Now the results. 29 different silanase producing fungi were isolated from 20 different samples. Among these, isolate X5 isolated from wood residue gave the largest zone of hydrolysis on xylan agar screening medium. Macroscopic and microscopic characteristics of isolate X5 showed that the isolate was blackish brown in color with cottony texture. The spores were brownish black in color and globose, while the hyphae were aseptate. 18S rRNA gene was amplified using universal primers and 1.8 kb PCR product was obtained. The PCR product was sequenced commercially, and the phylogenetic analysis showed that the isolate X5 was closely related to Rhizopus oryzae. Effect of 6 different media was studied on the production of xylanase, and it was found that medium M4 containing wheat bran, potassium hydrogen phosphate, and yeast extract gave the maximum production of xylanase. Effect of different carbon sources, glucose, fructose, sucrose, maltose, xylose, xylan, and wheat bran was studied on the production of xylanase. It was found that xylose gave the maximum production of xylanase, followed by xylan and wheat bran. Effect of different pH ranging from 4.5 to 7 was studied on the production of xylanase. It was found that pH 5.5 gave the maximum production of xylanase. Effect of different temperature ranging from 25 degrees centigrade to 40 degrees centigrade was studied on the production of xylanase. Maximum xylanase production occurred at 30 degrees centigrade. The amount of xylanase reduced when the temperature is lower or higher than 30 degrees centigrade. Effect of different incubation time was studied on the production of xylanase. It was found that Rhizopus oryzae gave the maximum production of xylanase after 96 hours of incubation. It was concluded that Rhizopus oryzae gave potent production of xylanase that can be used in different industries. These are the references that are used, and thank you very much.
Thank you so much, ma'am, for your constructive feedback and informative insights. Our next guest is Saira Safdar from University of Veterinary and Animal Sciences on the topic of ameliorative effects of lysorise extract against acute arsenic toxicity in Labiorhita. Ma'am, please give us insightful comments and valuable information about your topic.
Assalamualaikum everyone. My name is Saira Sapte, Fisheries and Aquaculture Department from University of Veterinary and Animal Sciences, Lahore. The topic of my abstract is ameliorative effect of liquorized extract against acute arsenic toxicity in Labiorhita. So the abstract is this study focused on the effect of arsenic exposure in Labiorhita and tested the protective role of liquorized extract, a medicinal plant known for the main bioactive components in liquorized extract possess antibacterial, antioxidant, anti-inflammatory, and the hepatoprotective properties. Added to 3 different diets at 0%, 1%, and 2% level prepared by mixing liquorized extract with the basal diet. 120 individuals of Labiorhita were divided into 4 groups. One group served as control and other 3 groups were exposed to arsenic 1 over 10th of LC50 for 28 days. And 0%, 1%, and 2% liquorized extract in their diets respectively. Results showed that the arsenic increased stress markers in fish such as urea, creatinine, antioxidant activities, and liver function enzymes. While also damaging tissues in the liver, gills, and muscles, liquorice helped reduce these effects in a dose-dependent manner. The group that 2% liquorice showed values close to the control group, suggesting liquorice extract can reduce arsenic toxicity and protect fish health. So the introduction is water pollution is a major problem, global issue that harms aquatic ecosystem, wildlife, and human health. So the heavy metals are a significant concern because of their toxicity to aquatic life. Arsenic is one of the most toxic metalloid in the aquatic system through both natural processes and human activities. Arsenic can pass through gill membranes of the fish and build up in different organs like skin, kidney, and the gastrointestinal tract. It can cause damage to the genetic level, oxidative stress, and cell death by disrupting mitochondrial function, increase the production of the harmful reactive oxygen species, and reduce the availability of the antioxidants in cells. So these days, medicinal plants are being added to fish feed as a natural way to help to reduce the toxic effect of harmful substances. Licorice is a medicinal plant from the Fabaceae family, and it includes has extensively used in pharmaceutical, food, and health industries, and have the bioactive compounds flavonoids, glabrin, glycinic acid, licorizine, and liquoride, which possess antibacterial, antioxidant, and anti-inflammatory and hepatoprotective properties. Objective of this study is to evaluate the protective effect of liquorized extract on oxidative stress and renal function in fish exposed to acute arsenic toxicity. To assess the protective effect of liquorized extract on histopathological changes in the gill, liver, and muscle tissue of the fish exposed to the arsenic. So the methodology is the research work was carried out at Fish Reading Unit of the UAS, Ravi Campus, C Block, Patoki, and the experimental species is Rahu. So the extraction of liquorized rhizomes was bought from the local market, washed with distilled water, samples were ground into powder, powder was soaked in methanol for 72 hours and then used rotatory evaporated and then extract was collected. Formulation of diet is 30 cp diet was used, ground and screened and then add liquorized extract, add water, make dough, place in meat mincer pellets, form, dry them and then store in the vacuum bags. Experimental layout is the three diets D1 with 0% liquorized, D2 1% liquorized and the D3 with 2% liquorized. The experimental design is 120 fish were randomly selected from the tanks and kept in aquaria. Fish were divided into 4 groups and each group were considered 3 replicates of 10 fish per aquarium and the experiment was conducted for 28 days. Fish husbandry laboratory is acclimatized for 2 weeks and then 10 fish was stocked in each replicate and adjust all the water parameters and trial was continued for 28 days. And the fish harvesting at the end of the experiment starved the fish for 24 hours and anesthesia the fish and the blood, sewer, liver samples were collected. And the analysis is urea and creatinine. The EDTA was utilized after taking the blood samples which are to be allowed to settle at room temperature after being centrifuged and then serum was collected. And then the Lifeline Laboratories and Diagnostic Center in Lahore was commercially available as kits to measure the creatinine and the urea. ALP, ALT, and AST were measured by blood taken, separate serum, and the kits bioactive diagnostic bed Homburg were used for serum liver function test for analysis. Antioxidant enzyme and tabars, their catalase activities, SOD, and the lipid peroxidation were determined by following methods. And the one we know was used for the statistical analysis and the Dunkel multiple range for to compare the means. So this is the histopathology of the liver, histopathology of muscles, and the histopathology of gills. The antioxidant enzymes, the highest value was measured at the T0 with the 0% liquorized and the lowest value for catalase and SOD were measured at the control. But the highest value were for MDA were measured at the T0 and the lowest value for control. But T2 with 20% liquorized is close to the control group. So the same liver function test ALP, AST and ALT show the highest value for the T0 with 0% liquorized and the lowest value for the control. And the renal function urea and creatinine show the highest value for T0 level and the lowest value for the control level. The conclusion in this study focused on the harmful effect of liver heat and tested the protective role of licorice. Arsenic increased stress marker in fish and the liver function test while also damaging tissues in the liver, gills and muscles. Licorice helped reduce these effects in a dose-dependent manner.
Thank you so much, ma'am. Your information was very good, and thank you for joining us. And our next guest is Hansa Mobin from University of Sialkot on the topic of evaluation of ectoparasitic infestation and associated risk factors in broiler chickens from District Sialkot. Sir, please, ma'am, please come and give us valuable remarks on your topic. Thank you so much for joining us and thanks to give us valuable remarks. Our next member is Ikra Mumtaz from University of Sargodha on the topic of effect of selected plants extract on enzyme activity of housefly. Ma'am, please give us insightful comments and thoughtful observations about your topic.
Hello and Assalamu alaikum. My name is Ikram Mumtaz and I am here as a participant of 8th International Conference on Applied Zoology. The topic of my presentation is the effect of selected plants extract on enzyme activity of housefly. As we all know that basically pesticides are used for controlling different type of pests. For example, you can say rodents or the fungi, herbs which are harmful, but we all know that they have certain type of disadvantages, like they can pollute the environment, they can kill the non-targeted organisms, they can also lead to biomagnification which can affect human beings, such as they can cause cancer and kidney disease. So we have to find an alternative for this. So biopesticides are basically derivatives of living organisms, for example animals or plants or microorganisms, and these are less harmful, less toxic, you can say, and biodegradable, so they are much more better as compared to the chemical pesticides. So in this thesis I have worked on my plant-based biopesticides, and because of their source, we can say that the plant-based biopesticides are mainly in the form of plant extracts or essential oils, or they basically act as a feeding deterrent, oviposition deterrent, and they even are less harmful and they don't act as a toxic substance. Target pest, basically the target pest that I chose for my thesis was the housefly, because we all know that houseflies are the vectors, okay, and they are responsible for carrying certain type of pathogens for different diseases like polio, diarrhea, cholera, etc. And if the vector is not there, if we can control the vector, we can control different diseases that are easily spread by them. So my hypothesis is the study which is performed to determine the selected plant extract, and those selected plants are Curcuma longa and Turkey-spermum emmy, which are basically turmeric and ajwain, and either they can be used as biopesticide for controlling the housefly, and what will be their effect on their detoxifying enzymes in the selected pest. So materials that are used for this purpose, or methodology that was used, you can see that there's collection of housefly, and then there was the collection of the plant material. After that there was preparation of the plant extract, which was done through the method of distillation, and at the end it was adulticide bioassay. So in this graph, in the results, you can see that the comparison of my biopesticide was done along with the chemical pesticide, which is chlorpyrifos, and we can see that in this graph the percentage mortality for chlorpyrifos is much higher, around, you can say, above 94% hai. Then there is turmeric. In turmeric, after the interval of, you can say, percentage concentration of circumlonga as it is increased, you can say the mortality also increases, but it goes up to 85%. But in case of Trachysphaermum mammae, as you can see, at 100% concentration, the mortality is up to 90%, which means that it is more effective as compared to the Curcuma longa extract. Next, there is the graphical representation of level of detoxifying enzymes that is occurring in the Musca domestica. First is the alpha esterase activity, in which you can see that Circuma longa and Trachyspasm ami are significantly affecting its level. Then there is beta esterases. Now you can see that the Trachyspasm ami has more significant effect as its level is decreasing below 2000 mm per minute mg. Next one is the monoexogenase activity. Again, there is a huge difference between circumlonga and Trachyspasm ami. Next goes for the GST activities, and again there is a much higher influence of ajwain, or you can say Trachyspasm ami, as compared to the circumlonga. Last but not least is the protein activity. As compared to the control group, you can see that the Trachyspasm ami has much more effect as compared to the circumlonga. Next, we compared the estimation or enzyme activity of these two specimens along with the chloropyrifos. Obviously, we can see that the chloropyrifos has much more influence or effect as compared to the other two. But in this case, there is a little difference between the enzyme activity graph of these two. You can say the extracts. As we go or move along monooxygenase activity and the GST activity, you can see that there is a little difference, but again the Trachyspasm ami has much more influence as compared to the Circuma longa. Next, there was done phytochemical analysis of Trachyspasm ami and Circuma longa, and their components were studies. The conclusion for this thesis, or you can say my studies, is that the biopesticides can be made from Secuma longa or Trachyspasm ami, and they are much more beneficial as compared to the chemical extracts in certain, you can say, chemical pesticides in certain aspects, as Secuma longa extract can cause mortality up to 71% with LC50 of 16.160, whereas for Trachyspasm ami it is around 84%, which means that it is more effective, it is more significant against the detoxifying enzymes of the, you can say, our pathogen or our vector, which is the housefly. Decrease in enzyme level. There is monooxygenases, total protein and esterases. Significantly, there was a decrease in their level. As for the GST, there was a positive response in chloropyrifos group. It indicates the chances of development of resistance in the housefly. For the future perspective, to limit the damage due to chemical insecticides, alternative must be made, and that alternative must be biodegradable, and it should not have a harmful impact on the non-target organisms. So I hope that much work can be done on these biopesticides, the plant-based pesticides, which are better. Thank you so much.
So our next guest is Mamoona Aslam from University of Sialkot on the topic of assessment of ectoparasitic infestations and associated risk factors in ducks from district Gujranwala. Ma'am, please give us valuable remarks as well as pertinent comments about your topic.
My name is Mamoona Aslam, Department of Zoology, and from the University of Sialkot. And the topic of my presentation is assessment of ectoparasitic infestation and associated risk factors in duck from district Gujranwala. Let's start with the introduction. Ducks are waterfowl found on every continent except Antarctica and belong to the family entity and order Inseriformes. Duck meat and egg are highly nutritious and preferred by consumers due to their flavor, nutritional value, and high content of amino acids. Raising duck is one of the most productive practices because of their meat and eggs, with the total meat production 4.4 million tons in 2013, an annual growth rate of 3.2%, and reaching the total 7.2 million tons in 2018, and the world waterfowl production is fast expanding business. Ducks are susceptible to a broad range of disease brought by bacteria, protozoa, helminthes, and arthropods. Ectoparasites are thought to be primarily culprit behind the bird poor living circumstances, reduced vigor, and development retardation. Ducks also consume a variety of aquatic creatures included fish, snails, earthworm, and water cyclops, which serve as intermediate host and become affiliated with parasite disease. Several external parasites feed on blood, which frequently result in anemia in birds. Here is the problem statement that stated that inadequate health management and poor hygiene practice can lead to spread of disease among duck population, resulting in decreased productivity. Here are the research objectives that stated that to check the diversity of ectoparasite in ducks from district Gujranwala, to investigate the variation in ectoparasitic infestation among different habitat type of duck from district Gujranwala, to check the effect of associated risk factors and ectoparasitic infestations. Here is the overview of research methodology, in which firstly the birds were examined for parasites. Parasites were collected from birds and then preserved in the 20% of formalin, then placed in petri dish, washed with the KOH, and sample were placed on the slide and observed under the stereoscope, and then statistical analysis were performed related to these studies. Selected species of this research is domestic duck that belong to the family Entidae and order Enseriform. The scientific name of this species is Platyrincus domesticus. Domestic duck are commonly found in Pakistan, particularly in rural areas where they are traditionally rare. These ducks exist various purposes such as providing meat. That is the study area map of district Gujranwala. In this area, the 8 different random sites were selected for collection of samples. Here is the pictures of bird examination, parasite collection, preservation, and identification. Following are the associated risk factors, and the questionnaire were developed to collect the data related to these associated risks. Statistical analysis were performed by using the software that were used are SPSS and PAST, and statistical tools that were used are t-test, one-way ANOVA, logistic regression, and Shannon index. Here are the results in which the 5 different species were identified from 8 different sites. Here is the bar chart that shows the clinical burden of ectoparasite of 8 different sites from both pet shop and domestic. Here is the diversity of ectoparasites, in which the 5 different species were recorded from 8 different sampling sites. Here are the bar charts that show the effect of associated risk factors in hygiene and feed type. These bar charts shows the effect of associated risk factors of habitat, and these are the regression models of associated risk factors that are feed and habitat and hygiene. Here is the pie chart that shows the overall percentage of ectoparasites that is higher in domestic than pet shops. The domestic percentage is 4% and the pet shop percentage is 16%. Here is the table of the diversity indices of 8 different sites. It is mostly focused on Simpson and Shannon index. Here is the graph of kernel density, which shows that the data distribution of the data is normal. Here is the conclusion, which stated that the 5 different type of lysis were reported during the studies, and the Anaticola crassicornis were in greater number in domestic duck. The effect of hygiene have great impact on ectoparasitic infestation. Future recommendation of this research is the relevant information gathered for this study will serve as the foundation for future investigation into the identification and management of ectoparasites in domestic ducks. These are the references. These are also the references of this study. And thank you. That's all about my today presentation.
Thank you so much, ma'am, for your astute remarks. Our next and last session guest is Ikra Mukhtar from University of Sargodha on the topic of effect of different feed and physiochemical parameters on the growth of Moina micrura.
Assalamu alaikum. I am Ikra Mukhtar from the University of Sargodha. Today I am here at the 8th International Conference on the Applied Zoology 2025. I am thankful to the organizing committee for giving us the opportunity to present our research work. My topic of presentation is effect of different feed and physiochemical parameters on the growth of Moina micrura. As we know, the main issue of the aquaculture is the high mortality of the fish hatchling due to the deficient and the inappropriate food supplement during the early life stages. Many fish species like the carnivorous fish hatchling and the shrimp hatchlings are lean toward the live feed because of their engaging free swimming way of behavior. There are many species like the Daphnia and which can be used as a live feed, but I prefer the Moina micrura due to the following reasons. It contains the essential nutrient and the vitamins. Moina has the protein content equal to the 50% of the dry weight. It is rich in the unsaturated fatty acids, which is good for the growth of the juveniles. The size is small and appropriate for the fish and the shrimp hatch. It has fast generation times and the short life cycles. Versatile to the different natural circumstances. Incorporate in the streams, ponds and the swamps. Adaptable in the fresh water and withstand the variety of the environmental conditions. This research might be helpful for the new collections of the data about the growth and the survival rate of the Moina micrura at the different feeding of the chlorella, cow dung, poultry waste and the yeast, and at the different water parameters like salinity, pH, etc. The aim and the objective of this research is the main objective of this study is produce the bulk quantity of the Moina populations of the culture of the carnivorous fishes to determine the effect of the different organic feed and the water parameters on the lifestyle and growth parameters of the Moina micrura. To study the relationship between the different organic feed and the water parameters on the survival rate of the Moina micrura. To evaluate the maximum long duration of the life of the Moina micrura at the organic feed and the water parameter. The experiment was designed at the Fish Biodiversity Hatchery Chashma at the district Mianwali. The first step in the experiment is the collection of the Moina micrura. The second step is the identification and the third step is the separation of the Moina micrura. There is the source of the organic feed and the other feeds. Organic feed chlorella can be cultured at the aquarium. Cultured at the Fish Biodiversity Hatchery Chashma and then dried in the shade and grind in the powder form. The cow dung and the poultry waste are collected from the local farmer of the cow and the poultry producers. The yeast was purchased from the medical shop of the district Mianwali. Maintenance of the water parameters. Salinity and the pH was maintained by adding the 1 gram per liter NaCl and 1 gram per liter NaCO3 sodium carbonate in the fresh water. The light was maintained by using the electric bulb of the 55 to 150 lux frequency. The third experiment was set up outdoor to use the diffuse sunlight. In the control group, water parameters were left unchanged. In the phase 148, clean and the neat glass beaker was used. Each glass beaker was filled with 1 liter water with recommended water parameter like the salinity 0 and 1 and the pH 7 and 8 for the no light, diffuse light and the electric light. Different organic feeds like the chlorella, cow dung, poultry waste and the yeast were used throughout the experiment. Initially 10 healthy and the full mature Moina micrura with neonates were put into the glass beaker to observe the total lifespan of the each Moina micrura for the 10 days. The lifespan parameters were noted after the 6 or till the completion of the experiment. The freshly hatched neonate of the Moina micrura from the each glass beaker was transferred to the phase 2 for the checking of the growth and the survival percentage. In the phase 248, clean and the neat glass aquarium was recommended water parameter. With recommended water parameter according to the phase 1 were used. In this phase, growth rate, total productions, and the survival rate of the minor microrhiza populations for the period of 10 days was noted. Recommended organic feed were given to the minor microrhiza twice a day at the rate of 0.03 gram per liter. In the growth parameter, the length and the weight of the minor microrhiza and the neonate was noted. For the length of the minor microrhiza, body was measured from the anterior side of the head to the base of the caudal spine with the help of the projection microscope, and the average weight of the 10 minor microrhiza and the neonate was measured. After this statistical analysis was done by the one-way ANOVA and the SPSS, the average weight soft. The process of the maturation or the initial reproductive age of the minor microrhiza at the various organic feed in the different water parameters was noted. The average lowest initial reproductive age of the minor microrhiza, which is the 48 hours at the salinity 0, at the chlorella food are used at the diffuse sunlight. The longest time of the maturation was observed 90 hours at the salinity 1 ppt and the pH 8 at the cow dung and the poultry waste feeding when the light was absent. There is some values which shows the results of this. In this study, to produce a bulk quantity of the minor microrhiza population, different organic feed in the different water parameters were used. Chlorella, cow dung, poultry waste, and the yeast organic feed were tested in the water salinity 0, 1 ppt, and pH 7 and 8. The best growth was observed at the chlorella feeding to the yeast and then to the poultry waste, but the lowest results were obtained at the cow dung feeding. As the freshwater zooplankton, the water salinity is 0 ppt and the pH 7 as compared to the salinity 1 ppt and the pH 8 was suitable parameter to attain the huge population of the minor microrhiza. It is noted that in the water salinity 1 ppt and the pH 8, minor microrhiza faced a stress condition, as a result disturbed its growth efficiency. It was also observed that early initial age of the maturation, age longevity, maximum number of the neonate per day, longer generation time, and the high survival rate of the minor microrhiza were also achieved at the chlorella feeding and with water salinity 0 ppt and the pH 7. The findings of this study indicate that among the 4 organic feedings, chlorella feed appeared to be the most superior feed for the culture of the minor microrhiza at the water parameter 0 salinity and the pH 7.
Thank you so much, ma'am, to joining us and to give us valuable remarks. And thanks to all of you for joining ICAZ 8th International Conference.